In today's video, we will look at an experiment called chromatography. We will show you using chromatography technique how to separate different color material in a food coloring. To do this experiment, we need certain equipments. Chromatography is done in two phases. Phase 1 is called stationary phase, where we prepare chromatography paper by loading different food colors to test. Phase 2 is called a mobile phase, which also means moving the colors on chromatography paper. Let's start the experiment. We have 4 different food colors: red, yellow, green and unknown. We will do a chromatography test on them to see if these colors are pure or they are mixture of different colors. We start with phase 1. We prepare chromatography paper by cutting it to the right size so it can fit inside the beaker. We then draw a straight line 2 cm above the base of chromatography paper using a ruler and a pencil. It is really important to use pencil and not a pen because the ink inside the pen may leak during the test and spoil our separation. Once we have drawn a straight line, we will mark 4 points 1 cm apart so we can place our 4 different food colorings on it. We are now ready to put our food colors on these points. We have initialed each point with the color that is being used. We have here red, yellow, green and unknown food colors. Using a glass capillary tube, we will place each food color onto the points on the chromatography paper. To place the food color on our chromatography paper, we touch one of the point on chromatography paper very lightly with a capillary tube filled with a specific color. This will load the color. We will repeat this step with the rest of the food colors. Once we have loaded all the colors onto chromatography paper, it's time for phase 2, which also means a mobile phase, which means moving these colors onto chromatography paper. To run the test, we need a beaker with tiny amount of water. Make sure the level of water is below the baseline or your straight line on your chromatography paper. Otherwise, it will spoil your separation. Now, we will place our chromatography paper inside the beaker and observe. Water will be drawn up on the chromatography paper by a capillary action. In few minutes, until water level has risen nearly to the top, you will see which food color is a mixture of different colors.